So we've spent the bulk of our time in this learning unit on lipid metabolism, talking about dietary lipids, which are really triglycerides. So taking those triglycerides in as dietary foods that we consume, and any excess food, any excess caloric intake is ultimately going to be converted into triglycerides or stored as triglycerides in adipose tissue. So the last little piece here is to talk about another really important lipid, and that's cholesterol. And so we're going to discuss in this video receptor-mediated endocytosis of LDL particles. But just as a review, if you haven't yet watched the video on lipid transport, I'd highly encourage you to do that. But we are going to be focusing specifically on LDL particles here. Remember, with lipids, we can't have them just free-flowing throughout the bloodstream. So whether it's fatty acids that are packaged with albumin, or we have all of these different lipoproteins, which are largely, we've got a phospholipid monolayer, so we're making these micelles, if you will, that have different proteins that are wrapped around them that increase their solubility and also provide directions for where those particles are supposed to go. And then inside, there's some semblance of cholesterol, usually as cholesterol esters. And then these yellow little spheres represent triglycerides. So specifically here in receptor-mediated endocytosis, we're going to be talking about LDL particles. So the key here is to recognize that LDL particles contain the lipoprotein ApoB100. So that's what this represents. That's the protein that sort of wraps around it. And that's going to be important because that ApoB is directing those particles from the liver to peripheral tissues. So we're going to talk about peripheral tissues that contain the receptor for ApoB100 and then how they're going to pull in those LDL particles into their cell to be able to utilize those cholesterol molecules and the triglycerides for their own use. So that is through a process called receptor-mediated endocytosis. So I'm going to walk you through this image first, and then we'll step back out and we'll sort of write some notes on it. So we are going to start at number 1 here, which really looks at that LDL particle. So it's drawn very simplistically here, just a simple cartoon representing sort of this lipid monolayer. And then we've got the cholesterol esters that are sort of inside. It's not really that the cholesterol esters represent that that monolayer really, but we've got cholesterol esters on the inside, and then here's that ApoB100 protein that's on the outside. Okay, so lots of these floating around in the bloodstream, and your serum cholesterol level, your LDL cholesterol level, is largely determined by the flux of these particles into and out of your bloodstream. So if we talk about what happens here, we're going to see that that ApoB100 is going to bind to receptors on the cell surface. So these little Y-shaped molecules here are receptors for the ApoB100 protein. And so what happens here is you're going to have receptor binding, and that's going to recruit these special proteins here called clathrin triskelions, and that's going to recruit them to the surface of that lipid bilayer. And that's going to facilitate the mechanical invagination that is going to bring in this particle. So we're going to invaginate and we're going to bring this in. This is essentially receptor-mediated endocytosis, is the bringing in of this vesicle. So we're going to bring this vesicle in here, and this is called a clathrin-coated vesicle when we have it in here. And what's going to happen then is we are going to fuse with an endosome. And a key piece here is to recognize that the endosome is going to have a low pH. Okay, it's going to have an acidic environment. And that is going to facilitate the dissociation of this protein from its receptor. Okay. And then the recycling of these clathrin triskelions back into the cytosol. And so this fusion here is going to facilitate all of these processes. So we're going to have these receptors then can find their way back to the cell surface. So they're going to sort of bud off in their own little vesicle, come back and fuse with the lipid bilayer. And so now we've got this endosomal particle, low pH, and we've got our intact LDL particle that's there. The next thing that's going to happen is we're going to fuse with the lysosome. So the lysosome has a high pH. Not only does it have a high pH, but it has lots of enzymes in there to facilitate degradation of biological molecules. very important to highlight that the lysosome, having these enzymes operate at a fairly elevated pH compared to physiological pH, allows them to make sure that if, for example, a lysosomal particle were to burst, the chances of those enzymes causing a lot of damage is less problematic because they're required to be at that higher pH in order to have their optimal activity. Okay, we'll revisit lysosomal particles later on. So what we have after we fuse these two together is we have something that's called a secondary lysosome. Those enzymes that are in there are going to break apart. Again, this is a protein here, so we're going to have all of these amino acids that we have here. We're going to be able to sort of break down all of the triglycerides into glycerol and their fatty acids, and so that's what's represented by this red here. And then we're going to have the cholesterol that's within this particle ultimately find its way into the cytosol. So a couple of other videos that we'll have that will highlight is the intracellular concentrations of cholesterol ultimately are going to affect a number of different pieces here. And we're going to see this here, that it is going to inhibit the synthesis of HMG-CoA reductase. And remember, that's an important enzyme that's responsible for the synthesis, the biosynthesis of cholesterol. And this makes sense, right? Feedback mechanism. If you've got plenty of cholesterol inside your cells, you don't need to be making it yourself. So you're going to inhibit that rate controlling enzyme. You're also going to decrease the synthesis of receptors. Again, if you've got plenty of cholesterol in there, you don't need to be putting more receptors on your surface to bring in more cholesterol. So this feedback sort of system works in so much of biochemistry to sort of regulate flux of biological molecules into and out of systems. Okay, so let's come back here and we'll take a minute and we'll just sort of review and then write these pieces down. So again, if you're, good at this point with what happened, you can fast forward through. I'm going to walk through sort of an animation in just a little bit for how this all happens. But let's take a minute and we'll sort of summarize these different spots. Make sure we write down sort of the relevant pieces of information. So really in steps 1 and 2, what we really had was that ApoB100 protein. So the ApoB100 protein that's on that LDL particle is going to bind to that receptor. Okay, and just to highlight here, that ApoB protein, this is not our little hat that we put on to sort of get our fatty acids into our mitochondria. But that receptor, again, looks like this, so identifying where those pieces are. And then we're going to have these clathrin triskelions are going to help with our mechanical invagination. Okay, so that gets us through to essentially having this vesicle inside of our cell. So steps 3 and 4, okay, are going to fuse with our endosome. That endosome having a more acidic pH, okay, and that's going to facilitate the release of our receptors from the LDL particles. Release of receptors from LDL particle. Okay. And then in sort of this, we sort of have a bifurcation of this pathway here where we talk about two different things happening. Steps 5 and 6 kind of highlight the recycling of the receptors to the surface. So we're going to have recycling of LDL receptors to the surface. And I want to highlight, and we'll revisit this later, that this is a key place where if this is faulty, if you have faulty recycling of those receptors to the surface, that means cells aren't going to bring in as much cholesterol as maybe they would want or need. And that's also going to leave higher levels of LDL cholesterol in the bloodstream. And so that can increase your serum concentration of LDL cholesterol. So faulty recycling can lead to elevated serum cholesterol. specifically LDL cholesterol, right? And so that is going to be a key place where, people who have sort of a genetic predisposition may have a situation there where they've got that faulty recycling. Okay, now this last little piece here that we sort of talked about, steps 7 and 8, is we're going to fuse with an endosome. So we're going to fuse with an lysosome. So fusing with that lysosome, we've got a more basic pH, so an elevated pH that we're going to have there. And that's key here because what happens here is we are going to have pH optimized enzymes are going to degrade proteins to amino acids, and then triglycerides, it's going to break those down into glycerol and then free fatty acids. Again, you can have those cholesterol esters as well. Remember we said a cholesterol ester is where you have that single OH group that's on cholesterol esterified to a fatty acid, and so you can make a cholesterol ester. And so we also can have esterase activity that's going to generate free cholesterol. So I'm going to have triglycerides, and our cholesterol esters are going to generate glycerol, fatty acids, and then cholesterol. Okay, and so we can see here just highlighting, we've got proteins being made into amino acids. We've sort of got those there. We are going to have our cholesterol esters, okay, are going to be made into fatty acids and cholesterol. And then we've got our triglycerides are going to be made into glycerol, and that also is a component for fatty acids. Okay, so that sort of takes us through what we have here, and then highlighting that this last important step that we'll expand on in another video here is when we've got these serum levels of cholesterol, or I should say cytosolic levels of cholesterol, that is ultimately going to determine what is happening. So number 10 is key, is because cholesterol levels that are in the cytosol, okay, will either activate or inhibit a variety of processes. Okay, and we'll revisit those in a little bit. So again, activation symbol with an arrow, inhibition symbol with sort of that little T symbol there. So we're going to revisit that on another slide. But what I want to take a minute to do is to show you a couple of videos, or actually it's one video, that is going to highlight this process and sort of animate it for us. I think it might be a little bit easier for us to understand. So they're going to walk through this video here, but just to orient you, here's our LDL particle. Remember, it is a micelle, so it's a phospholipid monolayer. Here is our cell surface that has our receptors. Here's those clathrin triskelions that are going to help with formation of that endocytosed vesicle. So not too much, I'll probably interject here. I'll probably just let this play.
In this example of receptor-mediated endocytosis, a cell takes in a particle of low-density lipoprotein, or LDL, from the outside. To do this, the cell uses receptors that specifically recognize and bind to the LDL particle. The receptors are clustered together. An LDL particle contains 1,000 or more cholesterol molecules at its core. A monolayer of phospholipids surrounds the cholesterol and is embedded with proteins called ApoB.
So just highlighting here, again, this is our cholesterol. I think they're trying to show that these are like cholesterol esters because they've got our cholesterol molecule that's fused to a fatty acid. And again, we've learned this as ApoB 100, so that's the specific ApoB particle that we're speaking of.
These ApoB proteins are specifically recognized by receptors in the cell's membrane. The receptors in the coated pit bind to the ApoB proteins on the LDL particle. The pit is reinforced by a lattice-like network of proteins called clathrin. Additional clathrin molecules then add to the lattice, which eventually pinches off a part of the membrane. This internalized vesicle is referred to as a coated vesicle. Inside the cell, the clathrin molecules dissociate from the vesicle and from each other, leaving an uncoated vesicle. Although not shown here, another vesicle then fuses with the uncoated vesicle, which causes the pH of the uncoated vesicle to drop. This drop in pH triggers the receptors to be released from the LDL particle.
So a lot that happened there. Again, they highlighted, and this is true, that those clathrin triskelions, they are going to disassociate before we fuse with that endosome. So we're going to have sort of that uncoated vesicle that's ready to fuse with the endosome. The endosome won't fuse unless we sort of get rid of that protein coat, that clathrin triskelion coat. And then we fuse with our endosome. That's going to drop the pH. And that change in pH, we've learned a lot about how pH can affect sort of interactions between substances, those non-covalent interactions. And so once those receptors are released, they're going to sort of disassociate themselves in their own little vesicle. And they're going to highlight here how they are returned back to the cell surface.
After the receptors are released, the vesicle carrying the LDL particle fuses with a lysosome. A lysosome carries enzymes capable of digesting the LDL particle into amino acids and fatty acids. At the same time, the cholesterol is released into the cytoplasm, and the cell either uses the cholesterol immediately or stores it for later. Meanwhile, the cell's LDL receptors are returned to the plasma membrane. Once in the plasma membrane, they can take in more LDL particles by receptor-mediated endocytosis.
All right, so that's a nice little video to kind of have you envision sort of what's happening there. We're going to have a couple of subsequent videos here. They're going to talk about what that cholesterol inside your cell is going to signal to do. And again, if you've got elevated levels of cholesterol, you're going to be suppressing your own biosynthesis of cholesterol and then also suppressing the synthesis of additional receptors, because that feedback loop indicates that you don't need to have additional cholesterol.